injury. So with that, we can add to this model that the inputs from the parabrachial nucleus to the CA do contribute to hypersensitivity, and that then we've established a sort of like a circuit and cellular level mechanism by which the central amygdala and the brain in general are modulating pain up and down in the brain. And with that, I'd like to thank the lab members that have contributed to the work, and just advertise that we have postdoc positions, and I'm desperately looking for postdocs, so spread the word. And I'd be happy to take questions now.
Wonderful, thank you. Applause all around. So I have a question in the chat from Kathleen, and I was going to start with, that's all right. She said, what a beautiful presentation, a very complete series of experiments. I'm fascinated by the balance between inhibition and facilitation within individual nuclei. This pattern has been found in multiple nuclei, including on and off cells in the RVM, inhibition and excitation cells in the PAG. I wonder your thoughts if you think these pathways are all connected and driven by or drive the CEA, or if you think they're all acting in parallel. Great question.
Yes, that is a great question. The short answer is I think they are all working together, but that the recruitment of the specific pathways will be dependent on the state of the animal. And I intentionally say state very vaguely, because I think it might be the emotional state, it could be the injury state, it could be anything state-wise that decides whether the animal or the person is focusing on the pain response versus the, something else that drives analgesia, stress or something important for survival that makes you decrease pain. And I think that all these different cells and circuits, they're capable of driving hypersensitivity and analgesia, but that their recruitment will be differentially mediated depending on what's going on with the animal, if that makes sense. And I'm dying to do in vivo recordings in the CEA to see if they match the classical definitions for the on and off neurons that have been described in the RVM and the PAG and all that. But I shied away from calling them that because the definition used by Fields and their colleagues is not exactly identical to what we see, but I really want to see if that's the case.
I'm going to just jump out of order a tiny bit here since Ted Price has a question related to that. Ted, do you want to jump in and add to that?
Yeah, sure. Really great talk, as always. So is there a lateralization of the somatostatin analgesic part, or is it just the pronociceptive part that's lateralized?
That's a great question. And everything we're doing is in the right side. We've been ignoring the left side. Ben Colbert at UT Dallas, he's been working on the lateralization a little bit more, well, a lot more. But I don't think that he's looked at the cell type specificity. It's one of the things that that we have. It's like the beginner level people that come to the lab. They're assigned to do lateralization recordings so that we can eventually figure out whether the properties are different, all of those questions that are important, but we don't know yet. Based on Colbert's results, though, I think that it looks like the right amygdala might be pronoceptive and the left amygdala might be anti-nociceptive. But I don't want to speak for his data because I'm not entirely sure that it falls within all the different models.
Thanks.
That's what I recall.
Tor, I know you have your hand, but I want to get to Briha Ansari has a question. Thank you for the talk. Does the response in amygdala neurons differ based on the type of nociceptors stimulated? for example, A delta versus C, mechanical, thermal, and chemical. That looks like you studied all of those different modalities, so different fiber types.
Yeah, that's another great question. And based on the early studies performed by Bernard in the 90s, it looks like it doesn't matter. And now with other, like the studies that we've done also, it looks like pretty much as long as it's noxious, it activates the central amygdala neurons. But no one has actually looked at the specific fibers. So I don't know specifically that, but I know it's not modality specific. And it's also going back to Ted's question, is independently of where in the body. It's like everywhere in the body, as long as it's painful or noxious, then the neurons are activated.
Makes sense. Tor, did you?
Lateralization is also on the right, even when the stimulus are, for example, visceral, which are not lateralized. So that's also interesting. Sorry, go ahead.
Thank you. Tor,
Sure.
did you have something?
Yeah, thanks. This is just a beautiful story emerging in the amygdala about how something new is added that isn't pain as usual, I guess, right? And I'm wondering about the sensory versus the affective properties of pain and whether they can be separated in the amygdala. And alongside that, I mean, sort of two more specific angles might be, how does the kappa opioid story factor in? And what about spontaneous pain-related behaviors or avoidance or other kinds of things? Yeah.
All great, important questions that the kappa opioid receptors, Porreja and Volker, they published a study on that, and they showed that the kappa opioid receptors in the CA are involved in this hypersensitivity. And in terms of the affective and somatosensory, that's what's driving me like incredibly nuts right now. It's so much work, and we are beginning to tackling it. It seems to indicate that not all this, while all the cells are capable of driving and all the circuits seem to be capable of driving the somatosensory component, it seems to be quite more specific with the affective comorbidities and the affective component. We've looked at unpleasantness, like formally induced aversion, and that seems to be cell type and circuit specific. It's too early for me to make strong conclusions, but definitely it's not everything that we modulate. And the same is true for stress-induced analgesia. It seems to be also cell type specific. So we're looking into it. I'm very interested in looking at all these different factors that are known to coexist with injury. In patients, we know that it's not like we have them sitting there and telling them, does it hurt, does it hurt? They have spontaneous pain, and it depends on many other different factors. And that's one of the lines of research that we're hoping to follow up, and we're starting to tackling them. But so far, everything seems to indicate that it's modality specific. I mean, that it's pathway and cell type specific for those components.
Thank you.
Thank you, Yarimar. Thanks so much, everyone, for your questions. We're going to move on now, just in the interest of time. I know we have one or two more. If you have time, Yarimar, maybe check in the chat and you could answer if anybody has further questions for her in the chat. Thank you.